8.6, we are going to look at how genes are turned on and off in an organism, because you don't always need them on to, if you always had them on, it would probably be bad because you'd be wasting energy and different nutrients and things like that that you take in. So first we're going to look at how prokaryotes turn on and off a gene by controlling transcription. This is a little bit easier to understand. There's not as many steps to it as what there are for the eukaryotes. So this is the steps involved. There is what's known as an operon, which is a sequence of like regulatory parts of a gene and the actual gene itself. So part of that is what's known as a promoter. Promoter is a DNA segment that allows a gene to be transcribed. Basically saying, here's where the RNA polymerase needs to attach. The operator is part of DNA that turns the gene on or off. An operon includes, like I said, an operon includes the promoter, an operator, and one or more structural genes that code for all of the proteins needed to do a job. So again, the promoter basically signals, here's where you attach. The operator is going to be the spot that definitely allows the gene to be copied or not be copied. So operons are most common in prokaryotes. The lac operon was the first example of gene regulation to be discovered. So that's going to be sort of what we talk about here. So the lac operon has 3 genes that code for the enzyme to break down lactose. So they were looking at how bacteria broke down the lactose. So the lac operon acts like a switch. The lac operon is off when lactose is not present. When it is present, the lac operon is going to be on. So down below here you see, here's the promoter right here. And that's going to be where the RNA polymerase attaches to. And it will start to travel down the strand of DNA, but it gets to the operator and you see that there is a repressor here. That repressor won't allow the RNA polymerase to continue on down the line. So this would be considered off. There's no lactose present, so that repressor is going to stay there, not allow the DNA or the RNA polymerase to continue on. Down below, you can see that there was lactose present right here. It attached to that repressor and caused it to fall off of the DNA. Once it did that, it allowed the RNA polymerase to go down and start transcribing the gene for making the enzyme to digest lactose. So that is considered on at that point in time. So for it to be off, whatever it codes for needs to not be there, and for it to be on, whatever it's going to be working on needs to be present. So in eukaryotes, the gene expression is going to be many different points. There are different sets of genes are going to be expressed in different types of cells. Transcription is controlled by regulatory DNA sequences and protein transcription factors. So you have this area right here, which is your promoter, again, where your RNA polymerase is going to attach. You have these transcription factors that are going to attach in this area called the TATA box, which it gets that name because it's a segment of the DNA that is repeating TA, 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 TA over and over again. So these transcription factors will attach there and basically fold up the DNA so it basically then signals for the RNA polymerase to attach at that point where it folds it up, and then it will allow it to go down. If you don't have these transcription factors attach, it's not going to allow for transcription to occur because it won't allow for the RNA polymerase to attach. So transcription is controlled by the regulatory DNA sequences and the protein transcription factors. Most eukaryotes have the ATA box promoter. The enhancers and silencers speed up or slow down the rate of the transcription. So each gene has a unique combination of regulatory sequences for it to be on or off. So like I said, it's a little bit more complex than what the prokaryotes were. RNA processing is also an important part of gene regulation in eukaryotes. The mRNA processes include 3 major steps. So when the messenger RNA copies from the DNA, it has stuff that it doesn't need. So those things that it does not need are called introns, and that's what you see right there and there. The things that it needs are the exons. So intron stands for intervening sequence, exon stands for expressed sequence. So what will happen is it will have made that copy, then it will recopy itself, and when it does that, it will basically pinch out those introns, the material that wasn't needed, and it will end up just having the exons in that final messenger RNA that's going to get sent out of the nucleus. So again, the introns are going to be removed and the exons are going to be spliced together. And it will also add a cap to the messenger RNA, and a tail is going to be added to it as well. So that is the end of the notes on how genes are turned on and off.